Hi and welcome to my OCR A-level biology revision session with me, Christine. So today's lesson, we're going to look at the cell cycle and mitosis, cell division. So the cell cycle always follows the same procedure and can be broken down into phases. So we're going to start with the first one, which is called interphase. Now, interphase is actually broken down into three different parts. So the first one is called growth phase, which is the first growth phase, so G1. Now, this is where the cell starts to grow. Now, as a cell starts to grow, it also requires the proteins necessary to ensure that it can move through into the next stages and to ensure that it is able to go through the process of dividing to produce two genetically identical cells. So the first stage with that G1 cell growth and protein synthesis, when we get to the end of that phase, what we have is a checkpoint which determines whether or not the cell is ready to move on to the next stage or whether it has to wait and be in what's known as the resting state. So the first checkpoint then, it's going to look for, is the cell the correct size? Does it have the right nutrients? Have the growth factors, i.e. the enzymes that are necessary for the next stage being produced? And also, is there any damage to the DNA? If there is any damage, then we don't want to then move on to the next stage, which is S stage and DNA replication. If there has been damage to that DNA, we do not want to replicate that damage. So therefore, that can result in the cell having to leave the cell cycle. So if all of these checkpoints have been checked off and they are all correct, then the cell can move through onto the S phase. This is where DNA replication occurs. Now, remember that DNA replication is semi-conservative replication. And if you haven't already, please do check out my video on DNA replication, where I go through all the different processes which will lead to you having the new strand attached to your old strand. So if the DNA gets replicated then, and that process has occurred, what then happens is the cell will move through into the G2 stage. Now the G2 stage is again another bit where the cell will grow. And again, we have another checkpoint here just before the next stage. So we need to check whether the cell has reached the right size. Has the DNA replication occurred without any errors? Or is there any damage to the DNA? And if these points haven't been checked off, then the cell cannot move through to the next stage. So G1, S, and G2 is all under the bracket of naming it as interphase. So interphase is the longest time in which a cell will spend its life in. The next part that we're going to move through to is called mitosis. So mitosis is important for growth. It's also important for tissue repair and cell replacement. So if there has been any damage to the cells or the tissue and we need to repair them or replace the cells, then this will be done through the process of mitosis. The other thing that mitosis is important for is asexual reproduction. Now, asexual reproduction can happen in plants, animals, and fungi. Not all animals. However, some animals can do asexual reproduction where they are producing offspring that are genetically identical to the parent cell or the parent plant or the parent fungi animal. So if we look at mitosis then, it's important to note that mitosis is actually broken down into four different sections. Prophase, the first phase, Metaphase, second, Anaphase, third, and then last, Telophase. So I always use my PMAT as my memory of which phase is actually happening. Is it prophase, metaphase, anaphase, or telophase? Now, mitosis is the nuclear division. This is the point in which the cell is going to have the nucleus divided so that we can produce two genetically identical cells. So to ensure that this is happening when it needs to happen, it's actually controlled by genes. So these are called proto-oncogenes, which are going to stimulate cell division. Or if we want to suppress the cell division. We want to reduce the cell division. That is controlled by your tumor suppressor genes. So you will learn more about these when you look at module 6 and you go on to learn about body plans. But it's important that we note this important role that mitosis has, and the production of these new cells needs to be controlled, and therefore those checkpoints need to be ticked off before it can move on to the next stage. So there is another checkpoint in mitosis, which is actually in the metaphase, in the middle. So in metaphase, we will have another checkpoint just to make sure that our chromosomes meet where they should. So let's just talk about these chromosomes then. So we know that during S phase, the DNA is replicated. We know that this is semi-conservative. So what we're saying is we have our chromosomes, and we are going to have DNA replication occur, and we're going to end up with chromosomes, but this time each chromosome is made up of sister chromatids. So we have to now note that we've got some new names that we need to use. A chromosome on its own is then replicated to produce chromosomes made up of sister chromatids. Now, the first phase in mitosis is called prophase. So during prophase, the first thing to happen is that the nuclear envelope and the nucleolus is going to disappear. Now remember, your nucleolus is the place where your RNA nucleotides are synthesized. Your nuclear envelope was containing your DNA inside the nucleus. So what we're going to do is this nuclear envelope and this nucleolus is going to disappear. So the chromosomes are now going to be free within the cytoplasm. Those chromosomes are also going to do what's called condensing down. They supercoil. So this supercoiling means that the chromosomes become visible. So we can see the chromosomes because they have condensed, they've supercoiled. This is where they are wrapping themselves tightly and pulling together all the nucleosomes. Again, something you will learn about when you look at module 6. The next thing that happens is your centrioles will move to the poles, and they will produce spindle fibers. Now, centrioles are basically bundles of proteins, and those spindle fibers are part of your cytoskeleton. So what they are going to be able to do is move the chromosomes around the cell, around the cytoplasm. So during metaphase, what happens is those spindle fibers will have attached to the centromeres. Now the centromere is the connecting point between the two sister chromatids. And what will happen is those chromosomes are going to line up at the equator. We call that the metaphase plate. So those chromosomes lining up at the metaphase plate is a signal to say that we are at the checkpoint, that they've lined up correctly, and therefore we can move through from metaphase into the next phase, which is called anaphase. Now during anaphase, those centromeres are going to separate. Because those centromeres are going to separate, those sister chromatids are now going to be pulled apart, and they're going to make their way towards the poles. Those spindle fibers are retracting back into the centrioles. So as they are retracting back into centrioles, because they were attached at the centromeres, as they pull those sister chromatids apart, what you get is this V shape in the chromosomes as they're being pulled. And that's what we can see when we look at our microscope slide. So if we're looking down a microscope and we're identifying root tip cells that have been squashed, if you haven't already, please do check out my video on preparation of microscope slides. These microscope slides are showing us the different phases. So we can see in the middle, we have one which is representing our metaphase, and down at the right-hand corner, we can see one of the cells which is going through anaphase, because we can see these V-shaped chromatids being pulled apart. Now, as we move through, the next phase is actually called telophase. And in telophase, what's going to happen is those chromatids are going to reach those poles. So the spindle fibers have retracted, and they've retracted all the way into the centrioles, pulling the chromatids with them. And once they have fully retracted and reached the poles, we now call the chromatids chromosomes. They have reached the poles, and that means that the nuclear envelopes can reform around the chromosomes, and therefore creating our nucleus or nuclei, because we now have two in the same cell. And the chromosomes will actually start to uncoil. So they're going to start to become less distinguishable because they have now reached telophase. And after those chromosomes have uncoiled, the nucleolus, the part which is for synthesizing the RNA nucleotides, can now start to reform as well. So we separate them into the four phases: prophase, metaphase, anaphase, telophase. However, it is a very smooth process that happens. So sometimes when you're looking at some of the microscope slides, the cell might not quite be fully in telophase. It might be early telophase and late anaphase, or it could be late prophase and early metaphase. So it's important that when you're looking for distinguishing features, that you note, can you see a nucleus? Can you see the chromosomes lined up at the equator? Can you see the V-shaped chromosomes being pulled apart? And can you see the nucleus reforming? Can you see two nuclei? So you need to identify these different features if they give you a picture in your exam, which they do love to do. So after the cell has gone through that process of mitosis, prophase, metaphase, anaphase, telophase, we now have two nuclei within the one cell. Well, what we want to do is we want to now separate and split that into two identical cells. So we need to split the cytoplasm, we need to ensure there are organelles in both cells, and we need the nuclear material to be evenly split. And therefore, this is done through the process of cytokinesis. Now, cytokinesis is done in two ways. In animal cells, it is done by what's known as a cleavage furrow. What happens is the plasma membrane is cleaved and pulled right down into the middle until it connects and the membranes are close enough that they can fuse together, and then the cells will separate to give you two genetically identical daughter cells. So in animal cells, because they only have a cell membrane, they do the cleavage furrow cytokinesis, where it's pulled together and then it can separate to produce two cells. In plant cells, however, we have a cell wall. So with plant cells, it's slightly different. What needs to happen is in what's known as the cell plate. Now, this is normally what was before the metaphase plate. At that same position, what's going to happen is Golgi vesicles are going to line up at the metaphase plate. So as these Golgi vesicles line up at the metaphase plate, those Golgi vesicles are going to contain the necessary enzymes so that the cell wall can be formed as the membrane starts to fuse together. Because if the cell were to just cleavage, what would happen is osmosis would occur and it would cause the cell to rupture. So it has to be a very coordinated process so that the cell wall is produced, because remember, in plant cells, the cell walls maintain that turgor pressure and therefore keep that stability within the cell. So cytokinesis is where the splitting of the cells from one which has the two nuclei into the two identical cells which have equal amounts of all the different components. So the other thing they love to do when it comes to the exam is throw a question at you with pictures. So they do expect you to be able to identify looking down a microscope or looking at some photomicrographs. So here we can see 4 pictures, and they will then expect you to be able to identify what stage you're looking at and also be able to give the description of what's happening. So prophase is R, metaphase is Q, anaphase is S, and telophase is P. And we can tell that because in prophase, we can't really see our nuclear envelope anymore. So R doesn't really show it, but we haven't yet fully condensed down our chromosomes. So that means that we're at the early stages of prophase in picture R. Q, we can see they're lined up as a big mess in the middle. That has to be metaphase. In S, we can see the V-shaped chromatids, so that has to be anaphase. And then in telophase, we can see two darkened sections at either side, so at either pole, that has to be telophase. So that's one type of question they could ask you in the exam, assessing that knowledge. Another thing they may ask you to do is give you a number of pictures, get you to identify what phase it is, and then ask you to calculate the mitotic index. So with the mitotic index, what you need to understand is when you look down through the microscope, you're looking at the number of cells in your field of view that are undergoing mitosis. And then you would divide that by the number of cells in the field of view in total. So the number of cells going through mitosis means prophase, metaphase, anaphase, and telophase. So you would calculate all the cells going through all of those phases and then divide that by the total number of cells that you can see in the field of view. So if I tell you that in the example that we have 45 that are in prophase, 8 in anaphase, 4 in metaphase, and 8 in telophase, and there are 325 in interphase. They would then expect you to do that PMAT, 45 plus 4 plus 8 plus 8. So therefore my mitotic number of cells is 65. I obviously have 65 plus my 325, so that gives me 390 total cells in my field of view. So I would then just divide those, and that would give me my mitotic index, which would be 0.17. So it's important that you understand that not only can you do it by looking at the information, they may do it to you in a roundabout way where they say your mitotic index is 0.35 and you have 25 cells going through mitosis. Therefore, what's the total number of cells? So you can rearrange the equation to work this out, and therefore that allows you to be able to apply the knowledge. So I hope you have liked this video, and if you have, then please do click the like button and subscribe to my channel. Also, do check out my revision platform, www.aiqchat.com.